Hello students and my dear friends. In this video, we are going to see how monoclonal antibodies are produced by hybridoma technology. Before that, we will quickly recollect the meaning of the terms monoclonal antibodies and polyclonal antibodies. The B cells gets activated by a variety of epitopes present in an antigen, which then produces plasma cells secreting different types of antibodies to these epitopes. So, polyclonal antibodies are a collection of mixed antibody molecules that react against a specific antigen, each identifying a different epitope. Monoclonal antibodies, on the other hand, are antibodies that are made by identical immune cells that are all clones of a unique parent cell. Monoclonal antibodies are single antibody species which bind single specific site. Whether polyclonal or monoclonal, these are used in diagnostic and medical fields. The monoclonal antibodies are produced by a special cell culture technique called hybridoma technology. These monoclonal antibodies were first discovered in 1975 by George Kohler and Cesar Milston. And for this discovery, they were awarded a Nobel Prize in the year 1984 in physiology and medicine. Why this hybridoma technology? Why not just inject the antigens into the mouse and harvest the plasma cells from their serum and produce antibodies by culturing them in cell culture media? But this method has one limitation, that is, the plasma cells have short lifespan and they can grow on the media only for a short period of time and hence cannot be cultured in the synthetic media for producing antibodies for a long period of time. Thus, this limitation can be overcome by hybridoma technology. How come? Let us see that. Plasma cell having short lifespan is fused with cancer cells. These cancer cells are referred to as myeloma cells. Myeloma cell is a cancerous B cell, that is, it has the property to divide indefinitely and are immortal. So, in the hybridoma technology, the plasma cells are fused with myeloma B cells to produce a hybridoma cell. These hybridoma cells have the property to produce antibodies as well as divide indefinitely and hence immortal. Now, let us know the procedure for hybridoma technology. First and foremost step is to introduce an antigen with a single desired epitope into an experimental animal, usually a mouse. This is done by injecting the antigen into the mouse intravenously. That is, using a syringe, the antigen is injected into the vein directly to the blood. Since the antigen is directly introduced into the blood, the antigen reaches the spleen. What is a spleen? A spleen is a secondary lymphoid organ. This antigen activates the B cell in the spleen, which differentiates into plasma cells. These plasma cells produce antibodies against the antigen. After a few weeks, the spleen is harvested from the mouse and the plasma cells are separated from it. Plasma cells are fused with myeloma cells using a chemical called polyethylene glycol. In this fusion step, along with hybrid cells, even other cells are generated. That is, the fused mixture contains fused plasma cells, fused myeloma cells, unfused plasma cells, unfused myeloma cells, along with the desired hybridoma cells. In order to select the hybridoma cells from this mixture, HAT medium is employed. The expanded form of HAT is hypoxanthine, aminopterin, thymidine. In this HAT medium, only hybridoma cells multiply, whereas the other cells, they do not multiply. These hybridoma cells are selected and cultured in appropriate cell culture medium to produce large amounts of antibodies. One day, antibodies will be used as magic bullets for medicine. These were the words of Paul Ehrlich. Likewise, these monoclonal antibodies are now being used to a greater extent in the diagnosis of many types of human and animal diseases or infections. And also, they are used for treating several diseases like cancers. Researchers in India and around the world are also trying to find treatment for COVID-19 using monoclonal therapy. To meet the demand for monoclonal antibodies, milligrams, grams, or even kilograms amount of monoclonal antibodies may be required. Therefore, to produce monoclonal antibodies, a bioreactor equipment is used. One of the most important step, the critical step in developing the monoclonal antibodies is the selection of appropriate tissue and cells and thereafter to culture them. Through genetic engineering techniques, genetically modified plant cells, genetically modified animal cells, genetically modified microbial cells, and insect cells may be used to develop antibodies. But among them, these genetically modified microorganisms gain more attention for development of monoclonal antibodies. So the field of microbiology is advancing in the production of monoclonal antibodies. So in the next video, we will see the basic principles involved in selecting hybrid cells using HAT medium. Till then, goodbye.